Hey guys, and welcome back to SciSci. In this lesson, we're going to be exploring PCR, or polymerase chain reaction. And again, this is going to be a two-part video. One with a lesson on the concepts that allow this process to happen, and then another part where we're in the lab showing you exactly how to do this procedure. And this is going to be similar to our gel electrophoresis video that we did a week ago. So let's get started. PCR, the general gist of this procedure, is allowing us to produce and analyze very small amounts of DNA and copy them to have millions or even billions of copies of this DNA. And so that allows us to have a large enough sample to where it can be studied in detail. Now, PCR is very common. It's used in medical labs, but also it has a wide range of applications that we'll be getting into a bit later in this video. Now, moving on into the concepts that allow this to happen, we have thermal cycling. Well, not exactly this type of thermal cycling. We have thermal cycling as in we're cycling different heat as well as cold, and through these cycles we allow processes to happen that cause these DNA to rapidly copy. In this process, we have our DNA. So you have our small DNA sample. We have a primer. And this is something that we've seen before, where a primer is attached to the DNA to allow us to attach more nucleotides to that DNA and create copies. So here you also have our nucleotides. And there's one more concept that we have, and that is the Tach polymerase. Now, the Tach polymerase is found in hot springs. So I've drawn a little hot spring on the side, but it's found in hot springs, and it's specifically found in the bacteria Thermus aquaticus. And the sole purpose of using Tach polymerase is that it can function at very high temperatures. And so when we think about thermal cycling, you have high and low temperatures, and Tach polymerase can function at that high temperature, and this kind of foreshadowing to the process we'll be talking about a bit later. Now, Tach polymerase is also useful because in the past, when this was first found, polymerase from E. coli was used to copy this DNA, and that required the polymerase to be added in each cycle. But Tach polymerase can actually survive the high heat and different temperatures, so it's a much more resistant and much more useful polymerase in this situation. So as a recap, you have our Tach polymerase in brown, our nucleotides in purple, primer in light blue, and then DNA in green. And this gets us to our first step, and that is denaturation. Now, denaturation occurs at 94 to 98 degrees Celsius, which is quite hot. I know it's not in Fahrenheit for some of you, but 94 to 98 degrees Celsius is 201 to 208 degrees Fahrenheit. And denaturation, if we remember our lessons on protein and DNA, denaturation is when the structure is broken down. And so as you can see here, our DNA is broken down into two different strands. So you have our double helix separating. And this is something we've seen in DNA replication. So it's a thing that we've seen before. Continuing on to the next step, you have annealing. And annealing occurs at 50 to 68 degrees Celsius. Now, when we look at this step, you can see that we have our primers now attaching to the DNA. And if we remember, when we look at our DNA, it goes 5' to 3', and it's actually opposite. So this side is 5' to 3'. And so that's why you have your primers on either side, and this primes us to attach nucleotides to this DNA and allows the Tach polymerase to attach to something to start adding those nucleotides. And you can see that we have nucleotides surrounding it, and this is kind of how the environment would be in your PCR reaction. Now, the last in this three-step process is called elongation, and this occurs at 72 degrees Celsius. So this is in between the two temperatures, and this is where the Tach polymerase functions. This is when the nucleotides are added to the primer, and you get 2 times the amount, or basically 2 times the initial DNA sample. And now you have that 3-step process. You have denaturation, annealing, and elongation. Now, you can see that there's a 3-step process of denaturing, annealing, and elongation, but there's actually a 4th step called a hot start that's sometimes added at 70 degrees Celsius. And as you can see, elongation, where the Tach polymerase operates best, is also at 70 degrees Celsius. So you can kind of assume that this step is used to activate the polymerase and get it ready for priming and have fast PCR reactions. But if we look here, 1 cycle, yeah, it produces 2 copies. But if you continue to 2, 3, you get 4, 8, continuing to 16, 32, and you have as many cycles is 2 to the x cycles. And so it's so even though you have 1 cycle produces 2 and 2 cycle produces 4, as you continue, you have 2 to the x amount. So it's an exponential growth, and that means that there's much more copies of DNA being produced as you have more cycles. So we've gone through these topics, we've gone through the process, and we've kind of reviewed DNA replication with our polymerase, but I think the most important step here is understanding thermal cycling and how that ties in with our polymerase. And now let's get into the applications. So there's a number of applications that span multiple different fields. You might question, how does copying a DNA sample allow us to, let's say, do sequencing? Although PCR allows us to simply copy DNA, if we have radioactive tags on each of these nucleotides, you can actually see the order through these radioactive nucleotides. You can see the sequence of the DNA through these radioactive nucleotides. So let's say you have a order that is A, T, G, C. A, T. Just keep it simple. Well, if we have a radioactive marker for each of the complementary bases, so let's say now you have a sequence using that radioactive labeling. Now, for example, let's take pathogen detection. Let's say you have a sample of DNA that does not contain pathogen. Well, when your primer comes to target the sample, it won't be able to, because the sample does not contain the same order of bases, the sequence that is correlated to that pathogen. But let's say you have a sample that contains a pathogen. Well, when you run these cycles and the primer copies this pathogenic DNA, then your pathogenic DNA largely outnumbers the other DNA. And you can see that there is pathogen in your sample. And so that can be used in a number of cases. You also have sequencing, mutation detection, cloning, GMO testing. And so again, you have a number of different fields that use PCR. And so it's incredibly important to understand this process. Well, that's our lesson on PCR. We've gone over thermal cycling, different steps that allow us to do this amazing procedure, and the applications of it. Now, in the next video, you'll be seeing a in-lab demonstration on how this process is done. Thanks for listening, and I'll see you guys on the next one.